Okay, so let's pick up for part two of this review. So again, we're going to kind of jump here in terms of putting these pieces together. Where we left last time, we were talking about passive transport, the idea of diffusion and facilitated diffusion, and osmosis. Remember that simple diffusion is limited for lipid-soluble molecules because the cell membrane is permeable to lipids. Lipids freely cross the membrane. Okay, so now let's put this together with some of the other ideas here. Okay. So, what your skin is. Jumping a little ahead so we can put these ideas. We've talked about the skin. We've talked about histology. You have the epidermis, which is stratified, squamous, epithelial tissue. It's really hard to write on the screen. Anyway, so stratified squamous epithelial tissue, which means we get layers and layers and layers of cells. Not only that, it's keratinized. Stratified squamous epithelial tissue. So there's layers and layers of dead cells on top of that. So you get all your dead cells and you get all your living cells, and each of those living cells has its own membrane, which is a phospholipid bilayer. Now they're squamous cells, so they're thin, flat cells. But still, phospholipid bilayers. We get layers and layers, and I can't draw this to save my life. Layers and layers of thin, flat cells. Okay. The cell membrane is freely permeable to lipids. So let's apply here. When the last kid that I sent to junior high getting ready for like this, like he's a fifth grader, to orientation at the school, and what I thought was going to be, here's what middle school is like, turned out to be like a 3-hour presentation on the dangers of fentanyl. So if you're not familiar with fentanyl, fentanyl is an opioid analgesic, synthetic. It's extremely potent. It's very effective in terms of its use as a painkiller. But as most painkillers that are also very effective, it's turned out that it's used illicitly. The thing with fentanyl is that very small doses are very effective. So it's very easy to overdose if you don't know the dosage. So using it illicitly is not a great choice. But anyway, so during this presentation, one of the other parents had a question about fentanyl in terms of drugging people, because she had seen on Facebook that people were like soaking rags in fentanyl and then like stuffing them under your door handles of your car, and then you would touch it and pass out from the fentanyl, and then they would take you and traffic you. So the police officer was very nice, much more patient with this question than I have been in the past. No, that's not how that works. The reason for that is because the skin is keratinized stratified squamous epithelium. In order for that medication to get through your skin to the deeper layer of tissue, so underneath this is where we have the dermis. Remember, epithelial tissue is avascular. So there's no blood vessels in epithelium. That means there's no blood vessels out here in this outer layer of skin. All the blood vessels are down here in the dermis. So in order for that medication to get through to your bloodstream, it's got to go through all of these layers of dead cells in the epidermis to get to the dermis. Can it do that? Absolutely it can do that. Can it do that fast? No. We do use fentanyl as well as other opioid analgesics in what's called a transdermal patch. Transdermal patch, the patch has medication in it. You slap it on their skin and you leave it there. And in the next, 2 or 3 hours, it will soak through and they'll have some relief. It will diffuse because what has to be true about it, if it's going to diffuse through these layers, what has to be true about it is that it has to be lipid soluble. If it's not lipid soluble, it's not getting in your bloodstream through your skin, period. Like, it's just not happening because you've got all these layers of cells, and unless there's a channel for it or something, and there's not, it's not going through there. Lipid soluble molecules can freely diffuse to the membrane, but if you've got layers and layers and layers of them, it's going to take a while. So, for instance, when you use that morphine patch or that fentanyl patch with a patient, you don't just slap it on them and then take it off. They have to keep it on there for a long time for it to be absorbed through the skin. We can apply other medications transdermally. We can use transdermal testosterone. In fact, testosterone that's available as a prescription to patients is typically sold as a gel. It's I can't remember the brand name of it, but it's applied to the skin. It's a gel, and it absorbs through the skin. Does it absorb quickly? No, it's going to take a while for that medication to absorb all the way through those layers. Beyond that, we talked about membrane junctions as well, the junctions between the cells. And one type of membrane junction we talked about is the tight junction. The tight junction is just what it sounds like. The cells are tightly held together. And in a tight junction, we're trying to keep stuff from leaking between the cells. One great example of tight junctions that we discussed is at the blood-brain barrier. If we want something to go from the bloodstream to the brain, we can't go in between the cells, we have to go through the cells. So for medications like fentanyl or morphine, or things like propofol, all of those have to be lipid soluble, or they would not be able to affect your brain, because they have to be able to go through a cell. So again, big picture here, lipids freely diffuse through the membrane. Lipids freely cross cell membranes. The big picture ideas here, the epidermis being stratified squamous epithelium, stratified squamous epithelium being for protection. Layers and layers of cells that are there for protection. So no, you can't just touch something that has fentanyl on it and then overdose. You're not going to touch something and then it's just magically absorbed through the skin that quickly. Are there things that will diffuse to the skin very quickly? Yes. Is it instant? No. If we want it to go into you faster, we're going to bypass the skin. So like when you want to administer a medication to someone very quickly, you put it in their blood. Okay. So there again, this big picture idea. And that is the sort of thing that I'm really looking for here, is your ability to put the pieces together. So that if I tell you that testosterone is a steroid that's sold to patients as a gel that's applied to the skin, what has to be true about testosterone? You need to say, oh, it has to be lipid soluble. If you're going to absorb it through the skin, it's going to have to be lipid soluble, nonpolar, hydrophobic, however you want to think of that. Lipids freely cross the membrane. Is it fast? No, it's not fast. Because by crossing through the skin, we have to go through all those layers of cells because the epidermis is stratified squamous epithelium. Okay, so there's that. Now let's back up again. The cells, go back to where we started, before I started putting pieces together. There are some simple things that you do need to know about cells, right? Past this idea of membrane transport. One, you need to know the difference between passive and active transport. Sorry about that. Passive and active transport. Passive, high to low concentration, no energy required here. Active transport requires energy, ATP. Active transport, we're going to go from low to high concentration. Look at those different types of active transport. You should be able to tell me that endocytosis is active transport or exocytosis is active transport. Primary active transport, the most important primary active transport that you need is that sodium-potassium pump. And I do need you to know that this is pumping 3 sodium ions out and 2 potassium back in. This is in every cell in your body. This is essential. This is extremely important for electrolyte balance, for water balance, and for the electrochemical gradient that we're going to need to talk about when we get to the muscles and the nerves. So I really need you to know the sodium-potassium thing. 3 sodium out, 2 potassium in. Okay. In terms of the organelles, no function. I don't care that you know how they do this, but you need to know that ribosomes make proteins. You need to know that mitochondria make ATP. You need to know the Golgi apparatus is responsible for shipping and packaging proteins and determining where they're going, either out or in the cell. It's like FedEx inside your cells. The rough endoplasmic reticulum you should associate with protein manufacture. The smooth endoplasmic reticulum has a bunch of different functions. None of them are protein manufacture. Basic function of the organelles. I'm not going to ask you like how the mitochondria pulls off aerobic respiration, but I am going to ask you about the mitochondria making ATP. And more likely than not, I'm going to ask you to apply this in some way. So for instance, if I tell you that cyanide is a poison that stops ATP production, you should be able to tell me that that's going to affect the mitochondria. I don't care that you know how that works, but what you need there is that mitochondria make ATP. If I tell you that, for instance, ricin stops protein manufacture, what makes proteins? Ribosomes make proteins. Membrane junctions, right? We talked about tight junctions, desmosomes, gap junctions. Tight junctions, the cells are tightly held together, like we would see at the blood-brain barrier, for instance. Desmosomes are like little spot welds in between the cells so they don't rip apart. And gap junctions are electrical connections, like we'd see at the heart. So cardiac muscle fibers, they're connected by gap junctions, so that electrical impulse travels from cell to cell. We talked about brain extensions. One sort of running thing that's going to come up repeatedly through the semester is the idea of microvilli. Microvilli increase the surface area of a cell. Anytime you see microvilli, you need to microvilli in the cell, it's there to increase the surface area because we want to move something across that cell. More surface area, more stuff we can move. So things that would have microvilli, like the lining of your intestines where we're absorbing nutrients. Things where we don't need microvilli, you know, the heart, the bladder, like that's, we're not absorbing stuff there. Let's see. Okay, so this is where I'm going to stop here with part 2. We're going to come back with part 3 and look a little bit more at histology and tissue stuff that you need, and then hopefully wrap up with part 4.